Hi, my name is Sarah Fenton. I'm a student in the undergraduate research program at North Seattle College, which stands on the traditional homeland of the Duwamish and Coast Salish peoples. My research project centers on diversifying the insulin production process. I'm delighted to have this opportunity to present this work here. This project is really the product of some really innovative teamwork from students in two different NSC undergraduate research program cohorts, and I'm humbled to have been a part of it. So I want to start out by setting the stage with some background on diabetes and insulin in America. Currently, there are 34.2 million people living with diabetes. That's 1 in 10 people in the US. And 20% of those people don't even know. If you look at the data for prediabetes, you'll see a picture that is even more grim. And when you start looking at the cost, it becomes apparent that diabetes is a public health crisis. In fact, 1 in 7 US dollars spent on healthcare is spent on managing and treating this disease. To put this in context, within the last decade alone, the price of insulin, a life-saving drug for diabetics, has risen anywhere from 93% to 325% in the US. Ironically, the patent for this drug was sold originally for a measly $3 because the scientists behind it believed that it needed to be a relatively open access drug. In 2018, a study out of Yale showed that 1 in 4 diabetics have rationed their insulin due to cost. Now, if you're a diabetic or you know a diabetic, you know that this is exceptionally dangerous. Most diabetics can't go more than 2 hours without access to insulin, which means that alternative methods for production are necessary. So this really takes us to where our project began. We wanted to know, what would it look like for us to produce our own insulin in a backyard or a basement with fairly basic lab equipment? And as a community college program, we had to work under some very specific constraints, including a fairly tight budget and a very quick experimental timeline. This brings us to our research objective, which was investigating the feasibility of producing insulin in the edible oyster mushroom, Pleurotus osteratus, using a novel polyethylene glycol mediated transformation process of the human insulin gene. So I want to share 2 distinct parts of our methods, starting with choosing the right plasmid for transformation, and then looking at how we went from mushroom to transformant. So we used 2 plasmids. Our first was the PAN7-1 plasmid, which was graciously donated to us by the Dutch DNA biotech. This plasmid was principally used to test our polyethylene glycol mediated transformation process in a fungal species, given its fungal promoter. Our second plasmid, which contains the human insulin gene, was the PCMV3C GFP Spark. A couple of things I want to point out. This plasmid includes a protein marker, the GFP Spark. This simply means that if a successful transformation occurs, then transformants will glow green under a UV light. Most important to this is that this area shares a common promoter, that CMV3 or that CMV promoter, with the human insulin gene. Now, this part was key for our design because it means that if we see the marker in our transformants, then we can suspect that more than likely the human insulin gene is also expressing alongside the GFP Spark. This essentially serves as a preliminary confirmation that our transformation worked. And then I just want to mention, it's notable that these 2 areas of the plasmid are not fused, which means that this can only be a preliminary confirmation. That GFP Spark presence is only preliminary, and it doesn't 100% mean that the human insulin gene is actually expressing. But I'll get into that kind of a little bit later. So I want to walk through our procedure at a glance from mushroom to transformant. We started out by culturing sterile hyphae from the fruiting body of our oyster mushrooms. We then cultivated mycelial mats on plates containing potato dextrose agar. Our next step involved digesting the cell wall of our sterile hyphae in order to isolate protoplasts. This was especially tricky because we had to do this without lysing the cell. Our whole project kind of hinged hinged on the success of this. Those cell walls function exactly how they're supposed to, and transformation without liberating those protoplasts just isn't possible. So this wasn't nearly as straightforward as we expected, and we ended up attempting this specific step over a dozen times unsuccessfully using a number of varying enzymatic types, concentrations, and exposure times. This was the classic research conundrum. For us, we just couldn't get it to work. And it wasn't until we changed our filtering process that we found some success. I've included a couple of photos of the process. In that first photo right here with figure 1, you'll see some pretty dense and well-formed hyphae. This is early in the enzymatic digestion. There's only 1 or 2 protoplasts visible that have been liberated. This second photo shows the same process a little bit further on in the digestion. You can see many different protoplasts, and then you can also see an artifact right here of sort of that shell of the cell wall, and a protoplast right here that is being released, which was quite a fortunate find, I think. And then this last photo just shows how we counted our protoplasts once we got our procedure to work. We used a hemocytometer specifically, and then we diluted to this specific optimized concentration. And that was kind of the preparatory step to doing the actual polyethylene glycol mediated transformation. This was about a 12-hour procedure. We followed it up with 18 hours of rest for our transformants, and then we streaked 2 different kinds of plates. So I want to move into our results and our discussion. The best way to do this is to compare the visible and the microscopic growth, and then also follow that up with some of our confocal analysis. After the transformation process, we tracked the growth of our untransformed protoplast as well as transformed PAN7-1 protoplasts and the PCMV3 protoplasts as well. So we grew all of these on 2 different kinds of plates, including a simple PDA plate and then our selection media, which combined PDA with hygromycin. After 5 days of growth, we started using a pen UV light to check for the the presence of the GFP. And then we snapped these photos showing visible growth on the left and microscopic growth on the right after 12 days. So on the top row, you can see with the simple PDA plates, we saw a lot of growth, and this was expected. Conversely, on the selection media, we didn't see any growth. And it wasn't until we kind of started looking under the microscope that we were able to see some growth, but certainly nothing to the extent that we saw in the simple PDA. Additionally, we saw no sign of the GFP marker each day when we checked with that pen UV light. And so at this point, we figured that our transformation had not actually worked. And it wasn't until we used some better tech that we were able to see that we were wrong, and that there actually was a lot of GFP. In this case, we used confocal microscopy at 450 nanometers. And this is what we saw. So once we initiated the right settings, this is what we saw on the computer screen, and it was a total surprise in the best way, primarily because there was a lot of GFP. Again, we know that any amount of GFP indicates that the transformation of the PCMV3 plasmid worked. Now, if you're asking yourself why you're seeing so much of it, we had similar thoughts. In fact, every single one of our samples showed GFP. Now, this doesn't negate our results, but it does indicate that there was some cross-contamination that happened somewhere along the way, likely in the late stages of the transformation or during our streaking process. Either way, we were exceptionally excited about these results. So I want to go into some of our remaining questions and our strengths and challenges to end. Because we saw so much of that cross-contamination or evidence of that cross-contamination, moving forward, we will be working on optimizing our transformation methods, specifically in how we streak our plates. Additionally, we are looking at how we confirm gene expression in a more concrete manner. We are currently working on protocols to use an ELISA assay specific to insulin and proinsulin. And this is exciting because it will tell us not only if the gene is expressing, but if it's expressing in a way that we want it to, specifically in how the protein is folding. Because we are testing for human gene expression in a fungal species, though, this has presented us with many unique opportunities and challenges in terms of putting those ELISA protocols in place and figuring out how to move forward on that. So finally, our strengths, we moved forward despite some of our constraints. We isolated our protoplasts, we produced transformants with the P insulin plasmid, and in addition, some of the challenges that we faced, again, that cross-contamination, and specifically our access to the lab given COVID-19 restrictions. That was specifically really difficult for us.